Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with LeAnn Whitney, Associate Professor of Oceanography in the Corning School of Ocean Studies at Maine Maritime Academy. If there is someone out there who likes phytoplankton as much as LeAnn does, I'd be stunned. We had a great conversation about her science path, ongoing research, and the value and importance of teachers and mentoring. LeAnn was one of this year's 5-Minute Genius speakers at the 2026 Maine Science Festival, and it was great to have the time for a deeper dive into her work. I've included a few definitions for some of the things we talked about in the show notes. Our conversation was recorded in May 2026. I hope you enjoy it as much as I did. LeAnn, welcome to the Maine Science Podcast. I had the great pleasure of introducing you and listening to you speak at this year's 5-Minute Genius event, which was, as always, for me, a giant amount of fun. And I know a little bit about you, but not a ton, just enough to have squeaked by in that event. I was intrigued to see that you're one of 6, if I have that right.
Yeah.
Yeah. So as someone who's one of 5, like there's not a lot of us who are from those big families anymore. So I think it does shape you, right? Especially depending where you are in the birth order, you see different things. Anyway, you're doing really cool work, and anyone who self-declares or at least has their daughter tag them as a phytoplankton nerd, what was it that got you into your nerddom, for lack of a better word?
Well, I just want to start by saying thank you for having me. I'm excited to be here and talking to you and kind of telling you about my journey. I mean, I'll say that I got interested in phytoplankton in high school. So I am from Maine originally, and I grew up in Machias, so a pretty small town. And I loved science. And I think by the time I was a junior in high school, I had taken all of the science classes that I could possibly take. AP classes weren't really that popular at that time. And so my high school biology teacher told me about a program. It's called the Keller BLOOM Program, which was offered by Bigelow Laboratory. At that time, I'd never heard of Bigelow, but it was like this week-long intensive ocean science experience. And they choose one junior from each county. And so she encouraged me to apply, and I applied and got accepted. And so that was my first sort of ocean science experience. All of my other science courses in high school were very traditional, right? Like chemistry, physics, biology. I think I had an anatomy class in there, but never kind of too focused. And so I went to Bigelow Lab and did this Keller BLOOM program and just really enjoyed it. I guess I would call it, I don't know if transformative, maybe that's overstating it, but it opened up my perspective on science and oceans and phytoplankton. So Bigelow Lab really emphasizes and focuses on phytoplankton research. And I don't know if I had heard of phytoplankton. I probably had, but I didn't really know anything about them when I attended this program. And so not only did I discover the importance of phytoplankton in the marine world, it was also my introduction to sort of the card-carrying scientist, right? Like people with their PhDs and doctorates. And that was not a path, or I didn't really have exposure to that until I went to this Keller BLOOM program and discovered that they were also like normal human beings.
So weird, right?
Yeah, weird. Yeah, right. And really were excited about the work. And I've been saying this for a really long time now, that like excitement is contagious, right? Like you can get excited about anything if the person that you're talking to is excited about the work that they do. And so I found them to be excited, and I just got excited about the world of phytoplankton as well. And so after that, my senior year in high school, again, having taken essentially all of the science classes that I could, that same biology teacher offered me an independent study on marine biology. And from, a pedagogy perspective, it probably wasn't like the best design class. We had one textbook that we shared between the two of us, and we just kind of worked our way through the chapters. But I just really appreciated the effort. And then that kind of led me to going into pursuing my bachelor's degree in marine biology at the University of Maine at Orono. And so that's kind of like how I got started in the marine science world.
Did you see the connections between your kind of foundational science classes and ocean science, or was it a giant leap for you? Do you know what I mean?
Yeah.
I understand why there's a kind of standard path that we teach K through 12 because you do need a foundation of things, right? But the downside to that often is unless you're around people who are in different fields, you have no idea what's out there. And this is true for everything, right? Like, I'm sure if you don't have a theater program in your high school, you have no idea that part of what you're doing in English class, for example, is something that could then be applied in another way. So I'm just curious, did it feel all new or was it something you're like, oh, that's kind of what they were talking about?
I think that maybe came more for me in college and having more lab-based classes, and then being able to apply the concepts that I had learned in chemistry, at least from my angle of research that I do, like the chemical principles into the ocean realm, but also into like a phytoplankton. Cells are just chemical reactions, right? And so it took me a minute, but I eventually learned that. So I think it came a little bit later when I was able to draw those connections between those foundational courses and what's happening in the ocean environment and what's happening in a phytoplankton cell.
So if memory serves, you and your siblings were the first in your family line to go to college.
Yeah.
And then you've kept going with a PhD. That is not trivial. And that is a different path if you don't have a family history of it. As much as I think it can be really hard to be the first in your family to go to college, I think it can be even harder to be the first in your family to go to graduate school because the context just isn't there.
Right.
I'm assuming you got encouragement. I'm just curious also how it was for you.
Yeah. I think if you looked at my trajectory on paper, it looks very traditional. And in a way it is, right? Like I went to undergrad, got my bachelor's. A short break, I took a year off and then went to grad school and got my PhD and then did a postdoc. It seems like a very traditional path. But you're right in that I wasn't thinking when I was young that I was going to get a PhD. Like I said, I don't even know if I really understood what that was and how you got from A to B. So I am the youngest of 6, so that's where I fall in that order. And so by the time I was in high school, particularly, the sophomore, junior years when you're starting to think about what college you might go to, my older siblings had gone to college. So that jump wasn't that difficult. Like I kind of knew I was going to go to college. I did really well. I worked really hard in school to get good grades. And so I felt like I was ready for college. And then when I was in Orono or at the University of Maine in Orono, I had some great professors who were encouraged. Like, I kind of feel like I'm more of a product of like the mentors and advisors that I had along the way. So particularly as I were in my last few years in Orono, I had a great research advisor and everyone has to do sort of their capstone project. And I did my capstone project on phytoplankton, of course.
Whoa, what a shock.
Yeah, what a shock. And that was really great. So my research advisor was going out on a research cruise during our spring break. And so he invited me to go on the research cruise. And that was a really great experience. And then when I graduated, I had applied for, a bunch of jobs and I wanted to stay in Maine. And so, there's limited options. So I'd applied for jobs, but wasn't getting any jobs. And so I reached back out to my research advisor and was just like, hey, do you know of anything? And he's like, yeah, I actually could use a part-time help. And so I got a research technician position in his lab. And that very quickly led to me getting another research tech position in another lab. So I was able to cobble together a full-time position and had things like health insurance and a retirement plan, which I also didn't really know that those things existed either. But, I had really great experiences in those labs, and they encouraged me to consider graduate school, which again was not on my radar. And because I was at UMaine, they're like, why don't you take a grad course? So I took biological oceanography in the School of Marine Sciences and enjoyed it and did well. And I think part of it is maybe just kind of lacking confidence. And that's where the mentors and advisors come in and just making me feel like I could do it. Again, because I didn't have like anyone in my family who had gone on to pursue a graduate degree. It was really having those people who could see in you what you necessarily couldn't see, which is like that I could do it. And so that encouraged me quite a bit.
Yeah, I'm constantly struck by how many really accomplished people I meet who have often had to push through imposter syndrome and sometimes still have to,
Yeah,
which I kind of get. But as an outsider watching, it's easy to forget that doing the work sometimes doesn't allow you to take a step back and say, oh, yeah, wait, I actually do know this stuff, right? Because from my perspective, watching, this was kind of my joke when my husband was in grad school. We knew a bunch of grad students, obviously. And I was like, I'm not doing that. You guys are crazy. But I was always struck by how hard everyone worked and still thinking they weren't quite doing it right. Seeing it from the outside. Anyway, I do think it's really interesting. So having the mentors and having the infrastructure of people who probably at their own point had their own imposter syndrome really helps foster that.
absolutely. Yep.
All right. So I'm going to jump forward a little bit and we can talk about your research some with phytoplankton,
Yes.
which I totally let you go without circling back to like, what was it about phytoplankton that was just it for you?
I think it was learning that the single-celled organisms like shaped Earth over like long periods of time, like geological scales, but also just like everyday life, right? That was sort of that Bigelow Bloom experience and then bachelor degree experience. And then my graduate school work was getting in even like nitty gritty detail, which is looking at their sort of molecular abilities and what genes do they have and what do they turn on and off in response to a changing environment. And so I think it was just like I was always kind of awestruck by what they're able to do and how they help shape the earth. And they never cease to amaze me in their abilities and what they're capable of doing.
It's so interesting. I hadn't thought about the fact that they are geologic time frame, but also current day. Literally until you said that, I was like, wow, yeah, that is actually really cool.
Right. I mean, the earth is oxygenated because of the early photosynthesizers, which was a photosynthetic bacteria. We call them cyanobacteria. You know, we phytoplankton nerds like to say about like half of the oxygen we breathe is produced by marine phytoplankton. And they're the base of the marine food web, right? So everything either eats them directly or indirectly. So they just play this like huge role. I mean, I don't study like harmful algal blooms, but I also find that topic like really interesting, right? Like we need them, but at the same time, from an economic and an ecological perspective, they can be harmful. So I just find them fascinating. Like there's always kind of something to learn about them.
All right. So let's take advantage of your deep interest in that. And you tell me what you're working on research wise. You can pick a project that you've done and completed. You can talk about what you're looking at now.
Okay. I will say that I have never completed a project. I mean, if a scientist says that they've like completed a project, I don't know.
No, that's a really solid point because I'm not enjoying lab work, which is how I knew I should not be a lab scientist. I'm used to being able to complete a project because that's the appeal for me. But you're 100% right. Like one answer leads to the next, leads to the next, leads to the next. So there's not an end point.
Never. Yeah.
How about this? Pick stuff that you're really enjoying figuring out.
Okay. So I'll talk about maybe the project that I was alluding to in my 5-minute genius talk. So it's kind of using more like kind of code speak and not getting into any one thing. But really, I would say I'm interested in what allows phytoplankton to kind of live life under limitation. And the ocean is constantly changing environment. I mean, you think of a scale of an individual cell that's, you know, just a few microns big, that their world around them is changing really rapidly. And the ocean's a big place and their food source, their nutrients are really dilute. And so I just find it really fascinating to think about how are they surviving in this limited environment? And maybe what are they using to survive that we don't actually know that they're able to use? My current phytoplankton research project is testing the ability of phytoplankton, specifically the eukaryotic phytoplankton. Cells kind of come in two floor plans, prokaryotic and eukaryotic cells. And so I'm looking at eukaryotic phytoplankton and was testing if they're able to use a form of phosphorus that we didn't think that they could use, that only the prokaryotic phytoplankton, the cyanobacteria, could use. And through some research that I had done looking at their molecular capabilities, I was seeing some genes turning on that made me think that maybe they can use this alternative form of phosphorus that we didn't think that they could. And so started doing some pretty simple lab work. Phytoplankton are actually relatively easy to grow in the lab, right? You give them some seawater, some nutrients, some light, and many of them will grow really well in lab conditions. And so through this research, I was giving them a form of phosphorus that we didn't think that they could grow and found out that they were able to grow. And so that then led me to want to know essentially what genes do they have that enable them to do that. And that is very much ongoing work because we know what the prokaryotic cells have. And that's what we can look for in the eukaryotic cells. But when we see some of that, they're also clearly doing things with genes that we do not know what they are. So you're kind of looking for the needle in the haystack, but you don't know you're looking for the needle, right? You're trying to find something, you don't know what you don't know. And I, as a scientist, really kind of like that hunt. In my 5-minute genius talk, I talked about rooting for the underdogs. And this is kind of the underdog story, right? We didn't think we can do it. They don't like to do it. The cells don't, but they do it a little bit. And so how are they doing it? And those are the questions that I'm trying to answer.
So why phosphorus? Why that in particular?
Yeah. So all cells need phosphorus, right? It's in our DNA and our proteins and our lipids. And it comes in many, I call them flavors, so many different flavors. And cells have a preferred form of phosphorus called phosphate, but there are pretty large regions in the oceans where phosphate concentrations are low. And so cells have to use alternative forms. And so that's what kind of led me down that path. It's what alternative forms are actually biologically available to these cells. And so if we can have a greater understanding of that, I'm still trying to figure out what is the ecological significance of this, right? So we're starting in the lab. Once we kind of have more ideas of what genes they're using, we can start to look for those in the natural world and natural ocean environments and see how relevant it is in those real environments. So is there an ecological significance story here? And I'm still working on that.
So I'm assuming this kind of research could only truly be done in the last 10 to 15 years because of the vast advancements that have happened in analyzing genes and how they work, right?
Yeah. So I started my Ph.D. work in 2006, and the first phytoplankton genome was sequenced in 2004. And so my Ph.D. advisor worked on that first genome project. The molecular revolution being applied to ocean science was pretty new and up and coming. And so I was sort of riding that wave. And so for sure, really only able to do, in the last 10 or 15 years. And it has changed so much over that time and sort of the scale in which you're able to kind of investigate the molecular machinery of phytoplankton.
So this must have vastly changed ocean research then, too. I'm used to having this conversation with microbiologists, right? Genetics. And I hadn't thought about how incredibly transformative it would be when you're looking at single-celled organisms and being able to compare the different kinds. What a whole new world.
A whole new world. Because if you just, go out in the middle of the ocean and scoop a cup full of water and look at it under the microscope, everyone should do that because phytoplankton are very beautiful to look at under the microscope. But so many of them, because they're so small, also look really similar. But it's not until you start to dig deeper, looking at their genetic abilities, do you really start to see that they are functionally very different. And that's the part that I find really interesting.
So they look similar at first glance, but there's all these vast differences in how they behave.
Yes, exactly.
How deep in the ocean can you find the phytoplankton?
So they need sunlight.
So it's going to depend, right, on where in the ocean they are?
Yeah. Essentially, we call it the 1% light level. So anything sort of above that depth, you will find phytoplankton. And then below that 1% light level, there's just really not enough light to support vast amounts of phytoplankton.
Does that make doing these research cruises easier, or it doesn't much matter because you got to go and do the cruise anyway? I mean, it seems like the sampling itself might be easier because you don't have to have tons of specialized equipment to do it from the perspective of deep down.
Yeah. There's equipment that allows you to, we call it like profiling the water column, from surface down to depth. So actually, while they are growing and dividing and metabolizing in the surface water, their biomass sinking to great depths of the ocean is also incredibly important, right? It's bringing that carbon and sort of feeding the deep, dark ecosystems of the planet as well. So people actually study phytoplankton, maybe what's growing and dividing up in the surface water, but people are also sampling deep, hundreds, if not thousands of meters deeper into the water column and seeing, we can call it marine snow, like what's sort of falling down into the deeper water column to get a sense of how much of that carbon is moving from the surface down to the great depths.
That is wild to think about. Yeah. I was going to say detritus, but that's not totally fair.
Well, it is. It is detritus, right? That's just like the dead and dying stuff that's sinking. I think a lot of people think about a whale carcass falling. And while that's really important, you also have phytoplankton cells that can get stuck to something and kind of become heavier, and that sinks, and that becomes a really yummy food source for bacterial metabolism as well. So they are truly kind of influencing the whole water column.
So as you try to figure out the phosphorus turning on, turning off kind of thing, are you looking at different species or are you trying to do one at a time? I mean, how does that look?
Yeah, one at a time, just because these experiments are challenging, right? You have multiple conditions and replicates, and I work as a pretty small team. So myself, my last few years, so this work was done at Bigelow, even though I've kind of been at Maine Maritime for the last 8 years. So I had a research technician who worked full time on the project, and she was really like the hands-on person. So a really small team, really a team of 2, right? And I was remote. And so did really great work. And so we do it one at a time, but multiple organisms. And it's really the comparative piece that allows us to start to gain some insight into how they are doing it. Like we can see this gene in this cell that's similar to this gene in this cell, and they're both turning on under these conditions. And because it's a gene that we don't know what it is, but it's telling us that it's important. And so we're starting to put those pieces of the puzzle together to tell a story. But we need to keep doing that because the information is key here, is having just more information.
I feel like this is a perfect representation of basic research at a level that I hadn't thought about enough. It is so easy to forget how critically important the ocean is to life on Earth. And then, you started talking about like the geologic time and the present time and how important it is. It is kind of stunning to think about how little we know.
Absolutely. The molecular work that we're doing is new, but like there are facilities that you can just kind of send your DNA and your RNA out and get data back. I don't want to say we're not doing anything like groundbreaking because that's not true. We're proposing new questions and answering questions and sort of a novel idea, but like we're not engineering new equipment. It really is sort of like a basic science that we're doing, but being applied to ask new questions. And for me, that's the type of research that I really enjoy. And then digging in the literature, it's like, all right, what do we know? And to try to glean as much information from those who have done this before me. And I really enjoy that part of it.
How many different phytoplankton species are there?
Oh gosh, Kate, thousands?
Yeah, that's what I thought.
I mean, the order of magnitude, I can't remember if it's tens of thousands or hundreds of thousands.
And how many are similar enough from the perspective, like it does it in this one, so it leads you to the next one, it leads you to the next one. Do you know what I mean? Like some species are very, very different from each other, so you wouldn't necessarily go to that. Like, I'm sure there's enough for the entire rest of your career. That's not what I'm asking. I'm just curious, like, what the cascade is.
Yeah, so I started looking in species that are naturally found in an environment where the types of phosphorus that I'm looking at are present in greater abundance. So like where they might just have adapted to growing in that environment. So I started there and then started with, all right, how widespread is this ability? So that was like a pico-eukaryote, like a green algae. And it's like, all right, can diatoms do it? Can dinoflagellates do it? And then those are kind of big jumps, but just kind of getting a sense of like, do these different functional groups, do they all have the ability? And then from there, it's like, all right, some can, some can't. how about this diatom species compared to this diatom species? So in the end, what I have found that like some can do it, some can't, and there doesn't necessarily seem to be an obvious trend. And like that this is a diatom thing and not a dinoflagellate thing, right? Like I haven't been able to find that pattern yet.
So how quickly do these cells evolve, like these critters?
That's a really good question. I'm not an evolutionary biologist.
So let me tell you why I was asking. With the phosphorus in particular, it seems to me if they evolve relatively quickly, that runoff from inland stuff is going to vastly change the phosphorus level in the water, right? So if they evolve relatively quickly, then that is why you would see it there. So that's kind of what I was asking. Like those spots, do they come from the same parts of the ocean from that perspective?
Yeah, I think that's a really interesting question. And like, I don't necessarily think about those lines, but I will say this is an ability that prokaryotic cells have, and eukaryotic cells evolved from prokaryotic cells. So it's more likely that some eukaryotic phytoplankton have lost the ability. We call them jumping genes, right? Like cells can kind of take DNA from other things. So maybe they've gained, but it's likely cells have probably lost it because of where they're grown. Like you said, right? Kind of where they're found naturally in the environment.
I wouldn't have guessed this, LeAnn, but you have made me excited about this. It is contagious, number one, but also I didn't appreciate how much there's to learn. Like, this is so cool.
Yeah, it's incredibly complex. I really can relate to the idea of being a lifelong learner. Like that these seemingly simple organisms are incredibly complex and we still have so much to learn about them. You mentioned imposter syndrome, and I still feel like that's like, oh, I barely know anything about them, right? And I've been studying them for, 2 decades at this point, probably longer.
But they've been around for millions and millions of years.
Yeah.
They have an advantage there, right? So last question, I totally glossed over that you're at Maine Maritime Academy, which unbeknownst, I think, to a lot of people, does have some really phenomenal marine science research going on. I feel like Maine Maritime in many ways is a little hidden gem in what happens, but I think in particular, the ocean research that's going on is far too hidden from view most of the time for a whole bunch of reasons. I'm not judging anybody. It's just how it is. It's not the immediate thing that people think about. How much are you able to foster and mentor your own students there? Because it's all undergrads, so it's a difficult-ish situation in that like turnover is much faster.
Right. I maybe digress from your question a little bit. I'll start it by saying like Maine Maritime is the place I feel like I was meant to be. And I say that because I loved being at Bigelow. In some ways, it still kind of feels like home to me, but it is just a research institute. And so the engagement with students, I was really missing. And I didn't necessarily know that until while I was a postdoc at Bigelow Lab. That's why I was there. I kind of felt like I needed to kind of stretch out my funds a little bit. I also felt like I needed some different experiences because I was starting to think about what is the next step for me. And so I started teaching for a summer as an adjunct at Southern Maine Community College. And my intention was to do it for a summer. It'll stretch my money out a little bit more. I'll get the teaching experience that I felt like I was missing. And I loved it. It was so hard. It was incredibly hard. It was a 5-credit microbiology course. I love microbiology. I didn't even know 5-credit courses existed. And it was a summer course. So it was accelerated. So it was like a ton of time in a 10-week period of time. Like my daughter was young, right? I was still doing postdoc work at Bigelow, but I loved that so much. And it really made me feel like in the classroom is really where I'm meant to be. And I loved it so much. Again, I was just intending to do it for one semester that I stayed there for an extra year. Like I taught that following fall and spring and would have kept going had I not got the job at Maine Maritime. But like that teaching experience is really what made me feel like I can do this, that I'm pretty good at it, and that it fulfilled me in a different way than sort of the applied research did. And so that is when I started to apply for more teaching jobs. I knew that that was the type of position that I needed, is one where I was spending most of my time in the classroom and that I could do research on a different level and be creative in how I did that. So that brings me back to MMA, which is we have great resources at Maine Maritime. Like we are on the water. I'm staring at it right now with a wonderful waterfront who is incredibly supportive. So if we want to get out on the boat, it's an email away. And so I love being able to do research in my backyard. So I am able to do research on a lot of different levels here at Maine Maritime. I do like grant funded projects, but on a much different scale that's very sustainable here. So I have several grants from like private foundations. We're not talking millions of dollars, but like work that we can do here and do with undergraduate support. And that's really enriching and fulfilling. And it's led me to a variety of research projects that I never necessarily would have envisioned myself doing. But because of where I am located, like in the Gulf of Maine and in Penobscot Bay, people are really interested in. So like several years ago, I had a lobster larvae project just because of where I am, right? Like this easy access to Penobscot Bay and waterfront support. And so once a week for a few months, I had myself and some interns. We went out and towed a really big net looking for lobster larvae, which are the most adorable things in the world, by the way. And they're not even a phytoplankton. And I'm saying that, right? So that was really fun. I have another grant looking at like PFAS in Penobscot Bay. And that's been constantly evolving. Like the questions that we asked 2 years ago are not the questions that we're asking now. And so that's sort of like that evolution and iteration of science that I also really love. And I am not the I think, my collaborator and friend here at Maine Maritime is the environmental chemist. And I'm kind of coming at it from like the biological perspective. That's really fun. And so those are sort of like the research projects, but I also try to be creative in how I introduce research and experiences into my classes that I teach. Some of my colleagues here at Maine Maritime, we've written grants to bring in really high-end equipment that is intended for use in the classes that we teach. So we are exposing students to really great ocean research tools and incorporating it into our projects. So things like really the flow cam, I don't know if you've heard of that.
I have heard of it, yeah.
But right, they're really amazing, right? Like imaging microscope. We have tools for doing eDNA and applying that into our classrooms. I have a project that I did a few years ago. I call it the Poop Project. So I did this in an elective that I teach, microbiology here at Maine Maritime, where I reached out to a local community that was having conditional shellfish closures because of fecal contamination. And they were curious about whose poop does that belong to that's leading to this. And so with my class, we got some water and just did some very basic molecular tools and found out maybe who the likely source is, like who it's coming from, what animal. And I told them and they're like, oh, I think we know. And that has like led them to investigate that even further, right? There's really like hands-on, but informative research and doing that in the classroom. And so I do research like that too, right? Kind of bringing it into my classes. And I really enjoy that.
That's a real full circle for you as someone who was inspired by a high school teacher to go try this thing out and fell in love with it, to be able to continue that with college students and undergrads. For those who don't have the luxury of knowing an academic closely, like I do, undergrads can be really hard because they don't know what they don't know, and they also don't know much. So there's a whole infrastructure that needs to be put in for undergraduates to do research. I also think as someone who studied chemistry in college, doing undergraduate research is really wicked important because it helps you figure out if that's what you want to do or not. And that is so important to figure out early.
And not everyone's going to get, like a summer internship to be able to have that like hands-on experience. I mean, I know when I was in undergrad, I needed to waitress during the summers, right? Like I needed to make money to help support myself through the academic year. I feel lucky in that the last several years I have been able to offer through my research grants internship opportunities for a few students at a time. But giving students research experience in the classroom, right? Because you're hitting a much broader audience. And I really enjoy that. And I think we do a pretty good job of that here.
I think you have this extraordinary place where you're able to think about this really cool basic research and how does the world actually work, and then also do the hands-on applied showing the connection for students, how those things work. So super inspiring. This has been a really fantastic conversation. The only disadvantage for us putting this out only on audio is that no one else but me gets to see your whole face light up when you talk about it. But that's the beauty of having my own podcast here, is that I get that. So this has been a ton of fun. And I really appreciate both your time and also the work you're doing. It's really fantastic.
It's been fun. Thank you so much.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by following us on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Lozelle. And the Discover Maine theme was composed and performed by Nick Parker.